All right. So sorry about the delay. Welcome, everybody. As you have said, my name is Karin Hoffmeister. I'm actually at the Blood Research Center in our Research Institute, which is now called Versiti in Wisconsin. I'm also professor of biochemistry at the Medical College of Wisconsin. So today's lecture will be called Glycans and Blood Homeostasis. This is a slide of hematopoiesis, and some of you may be familiar with this, some of you not. It is essentially, hematopoiesis is the formation of blood cellular components. So blood, if you look at it, is very liquid, but it actually contains a lot of cells, and besides that, plasma, and which is containing proteins and lipids. So this whole process, which is shown, and I took this image from Wikipedia, which is, I think, a fairly consistent representation of hematopoiesis. This whole process here, which is relatively complex, takes place actually in the bone marrow, in adults, in any adult person, any adult human. So on the apex, that's essentially the old version of hematopoiesis. We have the so-called hematopoietic stem cell. The hematopoietic stem cell kind of sits on top of all these cells, which will be deriving out of that one cell, which is always present in the bone marrow and essentially self-renews, takes care of itself. So it's always readily available if we need more blood cells to be produced. And the numbers are quite high, will be produced on a daily basis. It's 1 billion and more cells which are newly produced, and this requires essentially a very tight controlled mechanism, how all these cellular components know when to produce, where to go, and how many of these we are needing and at what situation. I will not focus, so essentially, if I go further here, so hematopoiesis is very broadly defined as 3 lineages. The erythropoiesis, which is the science of red blood cells, which is depicted here. So red blood cells are produced, and they are the oxygen carriers. The cell which essentially is necessary to, well, make us live and provide oxygen in any organ. There's lymphopoiesis, which is essentially establishing the lymphopoid lineage, and that is very roughly composed of T cells and B cells and natural killer cells, and they are kind of divided here in this very wide spread cellular components, the depiction of these several lineages which go out of a common progenitor here, or up here again a common progenitor. You can see that these cells are kind of very linearly derived, and they essentially are produced by various mechanisms which I don't have time to go into, so I just skip those, but this is obviously a very complicated and highly regulated complex. What I will focus today is the third cell which is produced. That's the myelopoiesis, essentially this part which is, well, not really depicted here. Myelopoiesis is the third component, which essentially consists of granulocytes, megakaryocytes, and macrophages. What I will focus today is on this one part here, which is there on the very left, which is the megakaryocytes and platelets. How do we produce these large cells in the bone marrow to essentially then produce what we call platelets or thrombocytes, which are a very important, however very small cellular component of the blood. So why focus on megakaryocytes and platelets? And I depicted here a couple of images why I think they really play an important role in many diseases, in many parts of our body. One important message is platelets are anucleated. So once this big cell is done, it spits essentially out platelets into the circulation, and it gets rid of any DNA, and most RNA would be able to discard it. Platelets are they have a half-life of 10 days in humans, so they live for a very short time only. We have a normal platelet count, which we maintain. Each individual has its their own specific platelet count, which is 150 to 400 per microliter. That is very well defined in each individual. And an increase, which we call thrombocytosis, or decrease, which we call thrombocytopenia, in platelet numbers can be associated with thrombosis or bleeding, respectively. And that's kind of depicted here. So if you bleed, you develop petechiae, which are these little red dots here. So you can see this very often, for example, when children present in the clinic, they will present with these features, which you can easily see in certain parts of the body. We also need platelets when we bleed, so we need to repair, essentially, we need platelets to avoid bleeding. And if we have an increase in platelet function or platelet numbers, we develop thrombosis. And again, we can have myocardial infarct, stroke, or deep venous thrombosis. So these are all complications if we do not maintain that steady state of platelet count. So platelets are, even in anucleates, they are more complicated than many people may know or may not know. They actually have not just the task to perform hemostasis, to stop bleeding, but they also contribute, and this becomes more and more clear, to certain functions of immunity. So on the left, and this is again taken an image out of a review article, depicts hemostasis, the steps of hemostasis. Platelets detect vascular breaches, so if there's a hole in the vessel, they will detect it, they will act on it almost immediately, even before any red blood cell, any white blood cell comes. They respond to endothelial alarms. They respond quicker than any other cells. They will form clots, so stop bleeding. They regulate vessel permeability, therefore, so this leaking of blood, this petechiae, what I just showed, doesn't happen if we have enough platelets. They seal vascular breaches during the leukocytes essentially transmigrate. They play a role in this. And they also establish actually our lymph during development. So they have tons of stuff to regulate. But they also play a role in immunity. They essentially can kill bacteria directly. They have bactericidal components. They can bind pathogens and essentially degranulate and then kill them. They respond to leukocyte alarms. They form nets, essentially what we call netosis. This is another immune response, what we have. We do have a leukocyte recruitment, so they recruit essentially, because they're the first responders, they will recruit all other cells, and they actually interact with leukocytes directly to further promote signals to repair vessels, but also to promote essentially immunity. So they're heavily involved in many of these processes, including innate immunity. So they are complex creatures, even not having a nucleus. So what causes side effects of thrombocytopenia? This is a long list, and I won't go through this. Obviously, bone marrow doesn't produce enough platelets. Cancer is very often involved. Different types of cancers can have vitamin deficiency. Certain therapies, such as chemotherapy and others. Platelets also can become entrapped in enlarged spleens, and there are multiple causes for this. So you just sit there in the pool of that spleen and disappear, and they are not visible to the circulation. We have immune thrombocytopenia, which is a broad term, and under that many, many things fall, and this is a very murky area of clinical area. We have for sure antiplatelet receptor antibodies, which cause thrombocytopenia. And I will touch on this a bit later. There why that glycans are actually involved in all this. We have viruses which are responsible, drugs. Many drugs actually cause thrombocytopenia. We have essentially diseases or such as sepsis due to gram-negative bacteria, which cause what we call disseminated intravascular coagulation. It's a very pathological process. All these cause essentially thrombocytopenia, and some of this we understand, some of this we don't. So causes of stromatopsis, again, there are various mechanisms involved, and that's essentially the signs of excessive thrombus formation. The complications, as I've pointed out, the venous thrombosis can be the deep venous thrombosis or renal thrombosis, and there are many more. Arterial thrombosis are just depicted two, myocardial infarct and stroke, which everybody knows. Most of us have somebody in the family dealing with this. The causes of stromatopsis are, again, many of them. And again, there is most of adults, over 80% in adults have actually a reactive stromatopsis due to infection, inflammation, which is chronic, tissue damage, etc. Some post-surgery following bone marrow suppression. So there are many causes for this. And some of them are clonal thrombocythemia, so essentially, again, the bone marrow itself produces too many platelets, and there are various diseases which are associated with this. And again, these are all complex processes. I will not go into this, just a list of them. So thrombocytopenia thrombosis therapeutics, essentially, I don't think they have that much to offer people. So hence there's, I think, in my opinion, a huge demand on therapeutic development. We have platelet transfusion, which is a process which is very important. Again, I won't touch on this, but there's much that has to be improved. I think there are thrombopoietin mimetics, and I will go into what thrombopoietin is in the next few slides. Corticosteroids, maybe heparin, and other antithrombotic drugs in the case of excessive thrombosis. We have chemotherapy, obviously, for underlying cancer or other diseases which are cured by chemotherapy. And oh my goodness, okay, am I? My thing is anyway. The therapy essentially depends on the underlying disease. Obviously, I'm frozen again. As I remarked to you, this is a very simple slide for something more complicated here, but what I want to depict is essentially something which I think is visually really important to understand the complexity of thrombopoiesis. And this is essentially a megakaryocyte which has been cultured in vitro from mouse hematopoietic stem cells. And what you appreciate, I think, here is, so this is this big, fat, gigantic cell which disintegrates into these cytoplasmic extensions, which are essentially, this is a very mobile, very complex process which forms these branched and elongated structures. And at the end of each of these structures, at some point, so all this will go into the bloodstream and become a platelet at some point. And the nucleus is essentially kind of, just discarded by the megakaryocyte, and it will end up in the bone marrow and will be displaced there. So this is a hugely complex process, and this is not an easy undertaking to understand this. And this is, again, just to show you the beauty and the complexity of this gigantic cell. So just keep this in mind when you follow the next steps. So thrombopoiesis, as I pointed out, is a process of platelet production. And this is a very simplistic depiction of what happens really in the bone marrow. Again, this is the hematopoietic stem cells and undergoes various stages until it becomes this polyploid, very large cell, which will send these cytoplasmic extensions into the circulation, and this is what we call then platelets. So megakaryocytes, they reside in the bone marrow. They are actually the largest hematopoietic cells, which can be between 40 to 100 microns. They undergo, as I said before, endomitosis, and they can reach up to 64 N. So they have 64 nuclei in this large, gigantic cell. Each megakaryocyte can produce 1,000 platelets. The human body actually produces, again, over 1 billion just simple platelets. So in the equation of whole blood production, these guys are actually not counted because it's 1 billion of cells again. And thrombopoietin is the major hormone that induces megakaryocytes to differentiate and to mature. So thrombopoietin is depicted, oh my goodness, here, up here, so essentially it's a cytokine which will act on all these, especially start with the hematopoietic stem cell and guide essentially these cell populations into producing this large polyploid megakaryocyte at a certain point. So we call this megakaryocyte differentiation, maturation, and then the platelet release. So there are different steps. Yeah. Oh, okay. So, so the last steps of thrombopoiesis, so essentially when the platelets are released, require an immense and membrane and organelle extension to form and release those platelets, and then releases the endoplasmatic reticulum and Golgi, obviously. And what this depicts is essentially one of these cells, of the megakaryocytes in different developmental stages. And what you see here is by Golgi marker shown in green that sits in the small megakaryocyte at the megakaryocyte level. When the megakaryocyte expands, this Golgi apparatus will become larger and then enlarges again. And at some point where this proplatelet release is taking place, the Golgi disintegrates into something what we call Golgi outposts. So they are kind of getting out in these proplatelets out here to produce platelets at a certain, and during this process, another very important point is that megakaryocytes package lysosome and other granules into packages to be functional. So they need all this process to essentially assemble one little platelet which contains a lot of components, including lysosomal enzymes, and quite a bit of this. As I said, the last steps of thrombopoiesis, or thrombopoiesis at all, really requires a huge amount of Golgi in ER involvement, and this requires then a huge enzymatic processing, which has to be tightly regulated. And so over the past few years, what has been identified are a couple of enzymes which are either residing in the endoplasmatic reticulum or in the Golgi. And this is a simplistic version of the Golgi depicted, but the enzymes, and obviously there are many enzymes missing, but the enzymes or proteins which have been identified to be involved in thrombopoiesis by GWAS studies, by large genetic studies, are depicted in red. So all these, which are depicted here in the ER, are actually involved in forming platelets. And there, by the end, in the trans-Golgi and medial Golgi, there are also a couple of enzymes which are clearly identified in mice and humans to be involved in thrombus formation, or thrombocyte formation, platelet formation. And what I will focus on today is the last steps, essentially the terminal cyanylation, how that affects platelet numbers and platelet production. And I will not go into the enzymes, but we know from these genetic studies that addition or regulation of the last step, so the addition of sialic acid to glycoproteins, is really an important step on regulating platelet release and platelet numbers. So all these enzymes, these glycogenes variants, which have been identified, either lead to thrombocytosis or thrombocytopenia. So there's a huge role for these enzymes to be involved in thrombopoiesis. So this is essentially depicting here a mass spectrometry of platelet surface glycans. So the glycans were released, and essentially what you can, so these are M glycans analyzed by MALDI-TOF. And what you, I think, can appreciate is that the platelets have a high amount of glycans on the surface, like other cell types as well. I think what is really clear here is that we have on platelets a variety of ending glycans, which is on the surface of these cells, from high nanostructures to really complex end-link structures, including poly-LACNAC structures. So this is essentially just to depict that platelets have a huge amount of sialic acid, a sialic acid-rich surface. These proteins are all heavily glycosylated. This is just a list of them depicting N-linkhuman glycans or Olin glycans. So we have a variety, again, of proteins expressed as in other cells, which are heavily glycosylated. Some of them are have higher N-link glycosylation sites, and some of them have lower, but all of these proteins are heavily glycosylated, hence they contribute to platelet function. The glycosylation contributes in most of these proteins to function, but also to platelet survival and platelet numbers. So what this depicts is essentially, again, a newer pathway of investigation, how we actually regulate platelet numbers on a daily basis via glycosylation. So again, this is our hematopoietic stem cells. Over time, stromopoietin will guide this hematopoietic stem cell differentiation and maturation into this megakaryocyte. And what we know is that the megakaryocyte releases platelets which are phylated, so they carry particularly, they carry Olin glycans, but they carry also a lot of the simplistic structure here, which is the LACNAC-phylated LACNAC structures. And what we have over time, and others have demonstrated, that this essentially that simple N-acetylgalactosamine is a ligand for a very potent receptor, and that receptor sits on hepatocytes. And I will go into this Asheville-Morel receptor in a bit in detail. So essentially, loss of phthalic acid mediates clearance or an interaction with hepatocytes with this lectin, which is called Asheville-Morel receptor. And once the decylated old platelets, so essentially when the platelet ages, removed phthalic acids are lost, it will essentially send a message to the Asheville receptor to produce de novo thermopoietin, and this will essentially close the cycle, act on the hematopoietic stem cell again. So essentially the bone marrow has a reporter from the liver to understand that we cleared senescent old phthalic acid-deprived platelets and will then de novo produce thrombopoietin. So that simple structure here is involved in recognition via this receptor, not just by aging platelets, but it is also involved in during inflammatory processes such as sepsis, for example, caused by Streptococcus pneumonia, and there are other diseases which we know. Essentially, these viruses or bacteria which express phthalidases or neuraminases will cleave phthalic acids, and this will lead to engagement again with the same receptor, the Asheville-Morel receptor. So, and this essentially that process, just by removing that decylated platelet, will ameliorate, at least in mice, will ameliorate the pathologic thrombus formation. So essentially, platelets are removed from the circulation, which has been decylated, and form then microthrombi in those microvasculatures, what we call the disseminating intravascular coagulopathy. And so removal of these processes will essentially rescue survival of the mouse, will rescue the mouse lifespan, and will ameliorate the sepsis procedure. Now, is that important? There's certain mouse models, but there's also enough human evidence, I think, in humans to support these findings. So the Asheville-Morel receptor is essentially has been identified, or I'll explain a bit more about how we came to be interested, or how investigation led into being really interested in this particular Asheville-Morel receptor of hepatocytes, which is a rather unusual receptor. So, do I have to use the mouse pad? Yes. Oh, okay. Sorry for moving the slides. I have to use a mouse pad, so I think I'm touching. Oh, right. Okay, but I have to use a mouse. And then,
you want to point it?
Yeah. So the Ashwell-Morel receptor is essentially a prototype C-type lectin. It's a type 2 transfer membrane protein. It has been actually discovered by Ashwell and Morel in the 60s and 70s. And the discovery came about that radioactively silated proteins circulate when you inject them into a mammal. Now, if you desilate or acylate these proteins and label them, again, radioactively, and then you inject them into a mammal, they will disappear extremely fast from the circulation. And now when they removed essentially galactose residues, so the 5-sialic acid, they removed galactose residues, that procedure actually prolonged the lifetime of these glycoproteins. And the other observation what these investigators have done is that the radioactive protein is sequestered in the liver. So all these acylated glycoproteins will end up in the liver, not in the spleen, not in any other phagocytic organ where we know we have macrophages, we have all other kinds of receptor or lectin-bearing cells. It's a calcium-dependent receptor, and hence, oh my gracious, it's a calcium-dependent receptor which has been purified from rabbit liver membrane, and hence C-type lectin is essentially referring to the calcium dependence. Again, the Ashwell receptor is a very complex receptor, not just by recognition of carbohydrates. What it does is it forms essentially complexes, very well individually, I think, especially species-regulated complexes. So in a human being, it's the Ashwell-Morel receptor is a heterotetramer. So it has 3 type 1 subchains and 1 type 2 subchain. So this ratio essentially only then will represent the Ashwell-Morel receptor. So no matter if you have one chain expressed somewhere or the other chain, it doesn't function. So the receptor is really dependent in the cell on that complex formation. So my suspicion, the funny thing was even the investigators have identified the subunits and they identified this receptor. Now, if they knocked out either the type 1 or type 2 subchain, the mice which have been generated didn't show the expected phenotype. They essentially didn't accumulate endogenous decylated glycoproteins, and hence this receptor was kind of, considered not functional until essentially the platelet studies came along, showing that actually not glycoproteins are the ligands, but the platelet is the ligand for this specific C-type lectin in the human being and as in mouse models or rat models. So I think the overall idea about or what the receptor plays a role is to regulate serum glycoprotein levels under stress conditions. So essentially, if we have too many platelets or we have too little platelets, this receptor receives these messages and then can produce de novo platelets or elevate also other serum proteins, which I don't go into here. So this is, again, a depiction of the receptor here. So it's a hepatic receptor, forms these homo and heterodimers, and this is very species dependent. So in the human, as I said, it has 4 subchains. In the mouse, it will have 3 subchains. But what is clear from this receptor that it actually prefers complex ending glycans compared to the to lower and complex ending glycans. Oh my God. Okay, and so it has also, as any lectin, is essentially has residues, which, by the way, it's clear that it prefers certain glycans. It's never specific for one glycan. But what it is essentially recognizing is terminal galactose residues and NS2 galactosamine residues on glycoproteins, especially on lipids. And it requires essentially to have a high affinity status. It requires to a certain degree these complex ending glycans, and the affinity increases quite a bit, 100,000 times higher when you have the complex glycosylation compared to the single terminal gal or GalNAc residue. So again, this is highly regulated by the complexity of the glycan, but also by the complexity of its expression on the cellular level. So there are two things which play a role in regulating the affinity of ligands, and that's on both parts, the receptor expression, but also what the cell which binds to this receptor expresses on the surface. Now, what are C-type ligands? I mean, C-type ligands come essentially in different forms, and that's a simplistic version of this. They are selectins, such as P-selectins, E-selectins, and L-selectins. They are the endocytotic receptors, and the Asheville-Morel receptor belongs to the endocytotic receptor part. We have the colectins, we have proteoglycans, which can also function as lectins. And C-type lectins, they're highly abundant, especially the carbohydrate binding region is expressed in many, many proteins, and it can essentially recognize protein carbohydrates, but it also has sometimes binding affinities to other non-carbohydrate ligands. It can be found, the C-type lectin fold can be found on more than 1,000 proteins, and one has to investigate protein by protein if that's really a carbohydrate binding domain. The glycan binding by the C-type lectins is always calcium dependent, hence the definition of C-type. It's a calcium dependent lectin. And that's essentially the calcium dependent because of specific amino acid residues that coordinate the calcium and bind to the hydroxyl groups of sugars. So the calcium is nicely in the middle of that carbohydrate binding domain assembled, and then carbohydrate domain, so it forms essentially the center where the hydroxyl groups of sugars can bind in that carbohydrate binding pocket. So it is relatively complex, and it has to be investigated protein by protein. But what is also very clear from the biology point of view that the C-type lectin fold is evolutionary and ancient, highly preserved domain, and it can be adapted protein by protein to whatever is needed for many uses, and very often this type of lectins is used for innate immunity responses. So it's a highly, well, I think, conserved receptor, but also highly adaptive receptor, depending on where it fits and which cell it fits. And so these C-type lectins are really, well, I think, any of these lectins carrying the carbohydrate binding domains should be looked at maybe as, well, somewhat involved in innate immunity responses, including the Asheville-Morel receptor I responded to, or I referred to. So the Asheville receptor is, as I referred to earlier, it is an endocytotic receptor that's depicted here as a trimer again. The ligands are internalized by clastrin, so it's a classical endocytotic receptor, internalized by clastrin-dependent pathways and delivered to early and late endosomes. So it is, again, a complex process where this carbohydrate ligands will be essentially processed in all these granular components in the cell. And once the receptor is essentially endocytosed, it will be recycled or degraded depending on which ligand it's actually endocytosed. and will essentially reappear on the surface. So on another note, the Ashwar receptor on the hepatic surfaces is highly mobile and essentially non-stop replaced. So it's a very busy receptor, and that essentially, I think, requires sometimes maybe an hour to half an hour to an hour so the receptor reappears on the surface. So it's non-stop being recycled and non-stop being put up on the surface so essentially to regulate platelets and possibly other innate immunity responses. The interesting from the drug development point of view, the interesting part about this receptor is the Ashwar-Morel receptor. It's relatively or highly specific to, to hepatocytes. So it's, is my iPhone or what? Oh, sorry. So the Ashwar-Morel receptor is highly specific for hepatocytes. So it is really, as the trimer or tetramer in humans, only expressed in hepatocytes. Hence, what many drug development pipelines have used it for, using this very simplistic lactosamine structure covered nanoparticles with these LACNAC structures as ligands. So they also know that essentially spacing between these two receptors on the hepatocyte is very important to be engulfed by the hepatocyte, by the Ashwar receptor in endocytose or engulfed. The space of these receptors is very important. So essentially drug developers use that very simplistic and very specific system to cover or to coat nanoparticles in many variants, whatever you want, essentially add drugs into those nanoparticles, use the spacing to increase endocytosis, and essentially deliver drugs into the hepatocyte. And it is a very, I think it is clinical, in the clinical, that some of these nanoparticles are really being clinically developed there in phase 3 studies, phase 2 studies, all kinds of, depending where they are. But they have successfully used that system, for example, to deliver certain drugs or certain RNA particles or mRNA particles to kill hepatitis C viruses, to kill hepatitis C viruses. So it is essentially a very appealing, from that particular simplistic structure, it is a useful structure as a ligand for that particular receptor on hepatocytes. And so one idea I just want to leave it at is, which you can carry around and maybe think about when you get older and have use for this. You could envision that some of these nanoparticles develop in an efficient way could be maybe used as a new TPO mimetic. So TPO mimetics today are produced in insect cells, in some weird non-human cell type, and they have their own problems. They are working to a certain degree, but you could envision that generating a mimetic could actually stimulate stromopoietin production, the endogenous stromopoietin production, and then be a useful Ashwar-Morel mimetic. Further on that note, there is essentially this receptor, as I said, is a very complicated receptor. And so Amgen, who is one of the big drug developments in cardiovascular drugs, they sequence, in order to search for new drug targets, they sequence over 2,500 Icelanders. So it's an Icelandic population, which is genetically a very well-defined population. And in this population, they found variants, and they will impute it then in this Icelandic GWAS population. And what they have found is essentially a rare non-coding 12 base pair deletion in the Ashwar ASGR1, which is the type 1 chain of this receptor. What this has essentially led to is the loss of function of the receptor. And these individuals with variants have lower non-HDL, so essentially they have a lipid phenotype, if you wish. They have lower cholesterol levels. They have an increase in alkaline phosphatase. But what is really interesting, if you look into the drug development of Amgen, that the cardioprotective effect is one of the biggest they have seen and cannot be explained by just those cholesterol levels, including there's a much longer or much higher survival of these individuals having just a simple variant of the Ashford-Morel receptor. So Amgen actually developed an inhibitor using the knowledge of the deletion, and so forth. They developed an inhibitor called AMG 529, and a phase 1 clinical trial has been initiated and I think by now completed. So again, a simple hepatic receptor can be used as a drug development tool if you understand what it actually does or what it doesn't do. And that is, I think, another example how we can leverage T-type or lectin receptors, not just glycans on the surface, but also carbohydrate binding receptors to develop drugs. Once we essentially employ genetic studies, but also biochemical studies, because again, this receptor is much more complicated, so I am not sure that Amgen will really follow through with their drug, because I'm not sure they understand the full biology of that system. So back to platelet biology and a bit more how platelet numbers are regulated. So I have to just depict the very simplistic platelet biology. So what platelets do, as I referred to in the beginning, platelets are packaged with granules. So granules, and there are a variety of granules. We classify them in mostly 3, maybe 4, which is a new coming up granule. Alpha granules, dense lysosomes, and Golgi-like granules. So alpha granules, they contain P-selectin and all kinds of other clotting factors. They're essentially packed with clotting factors and growth factors. Dense granules, they are packed with adenine nucleotides and serotonin, so they're kind of the energy carriers. But they're also packed with lysosomes, and these lysosomes contain quite a bit of galactosydases or glycosidases, including salidase type 1 or new one, beta-galactosidase, beta-hexosaminidase. And what they platelets do is upon activation, so once they hit a wall, a vessel wall, which is damaged, they will release this granular content, including these lysosomes. So this is another powerful, I think, another drug development pipeline. What I want to what I want to emphasize is that all these granular contents, including lysosomes, can be thought about as something anti-thrombotic or regulating platelet count. So what we also know is that thrombocytopenias, they're often mediated via specific antibodies which bind to platelet receptors, and then we assume there's a platelet clearance via the spleen. However, again, newer insights into all this, it's not that simplistic. We don't just bind that antibody and clear via spleen. As I referred to, platelets actually have salidases. So certain antibodies will bind to receptors and induce release of salidases. This will lead to a cleavage of salic acid and then recognition by galactose receptors, including the Ashford-Morel receptor. So do bacteria viruses. So salidases from bacteria will deplete platelet surfaces quite efficiently. That is essentially including HIV viruses, influenza viruses, Trypanosoma cruzi viruses, and all kinds of bacterial viruses, including pneumococcus, streptococcus pneumonia. And so what is essentially becoming, again, more and more clear is that inhibition of that salic acid loss using salidase inhibitors may be actually a potent way to ameliorate either platelet depletion via the specific antibodies here, and possibly by bacteria and viruses. So what is pretty clear that the FDA-approved inhibitors, which were developed actually for influenza virus solidases, such as zanamivir or Lenzar, which is administered by inhalation. There's oseltamivir, such as Tamiflu, which is orally given to patients. This is a newer phase 3 clinical trial. I think in Japan it's already approved for treatment. All these drugs have been essentially developed to more selectively or not selectively inhibit neuraminidase or solidases from influenza A and B. But however, these drugs are given essentially based on when patients come into the clinic, they are given to patients when they have the diagnosis of influenza is suspected. They are giving Tamiflu orally, and some of these patient populations that have been identified, they had thrombocytopenia, which is very clear that some of these patients having the thrombocytopenia of unknown causes actually improve by just being given those virus inhibitors, solidase inhibitors, such as Tamiflu. The same for thrombocytopenia in patients with sepsis, bacterial sepsis. It ameliorated the Tamiflu administration, actually ameliorated thrombocytopenias, and that has been done in small clinical trials now in sepsis patients, and there's now one clinical trial which has been opened for immune thrombocytopenias to essentially get a proof of concept. So that's the clinical trial which is right now out at the NIH level. So Tamiflu will be administered to patients with diagnosis of immune thrombocytopenia. So with this, I think I'm done, and I apologize for what do I do now?